Hello, this is Module 6, and in this module, the first video will discuss gene linkage, chromosome mapping, and those are the main topics of this module. So I wanted to start out by showing the outline of topics for this module, and in the first video, we're going to tackle the first two of these. How do linked genes behave in meiosis, if they're on the same chromosome? How do they behave, and how is that different for genes that are unlinked? So we're going to look at our basic review of unlinked genes as seen by Mendel, and then we'll compare that to the behavior of linked genes. And the main way to do that is through the standard cross to check whether two genes are linked or not. So that's the other main topic for this video. All right, for unlinked genes, this is the situation that Mendel saw in whenever he did a dihybrid cross. And based on the results of his dihybrid cross, Mendel derived his fourth law, which is the law of independent assortment. So that's the, I want to review that briefly in a video, which I will start in a second. Before that, though, just a few more points about independent assortment and what happened after Mendel was rediscovered. In the early 1900s, geneticists realized that organisms had multiple chromosomes that carried genes. And however, they noticed that the number of chromosomes was relatively small and the number of genes was relatively large. So they realized that all chromosomes must be carrying large numbers of genes. And therefore, that many genes must be linked together on the same chromosome. So they began to question how genes that are linked are passed on and how that's different for genes that are unlinked. So let's take a look at the situation for genes that are unlinked. And this is what Mendel saw and how he derived the law of independent assortment. All right, this module is about gene linkage. And gene linkage was something that Mendel never saw. Whenever Mendel crossed genes for two different traits, that's the dihybrid cross, the results he saw illustrated the fourth principle of his, which was the law of independent assortment. The law of independent assortment states that alleles of two different genes sort independently of each other into gametes during meiosis. So I wanted to start by reminding you of the general cross that he used, the results that he saw, and also some of the terminology that we're going to be using a lot going forward. All right, so this is the typical dihybrid cross that Mendel carried out. He started out with a parental generation where two parents, let's say male pollen contributing parent, was homozygous for two dominant traits, and the famous one we've been using is P color and P shape. So this is a pure breeding parent that has yellow and round P's. And in his dihybrid cross, he crossed it to a pure breeding strain that differed for those two traits, pure breeding green and wrinkled. Yellow is dominant to green, round is dominant to wrinkled. Okay, so when he crossed these two strains together, he got an F1 that was heterozygous, one chromosome coming from the mother, maternally derived, one chromosome coming from the paternally derived, the pollen contributor. Right, so for the P color gene on this chromosome, heterozygous, it's carrying one allele of each. The chromosomes are in duplicated form, replicated form here, so they have two sister chromatids that are identical. All right, the gene for P shape is on a different chromosomes. They're unlinked. That's what unlinked genes are, on different chromosomes. And heterozygous again, one allele coming from the female parent, the other allele coming from the male parent. Okay, when this double heterozygote undergoes meiosis, the law of independent assortment states that the alleles from this gene, P color gene, are going to go into gametes independent from the alleles on this gene, P shape. And Mendel didn't know that, the reason why that is, but the reason is because these genes are on different chromosomes. How this chromosome behaves in meiosis is completely independent of how this chromosome behaves. They're going to assort independently of each other into gametes. What that means is the gametes that form from meiosis of this double heterozygote will have all four possible combinations of one allele of each gene. So those combinations are two dominant alleles, both paternally derived, dominant for yellow, recessive for wrinkled, recessive for green, and dominant for round, or both recessive traits, green and wrinkled. These gametes will form in equal frequency, 25% each. The reason Mendel knew that was because when he crossed two double heterozygotes, and this is this cross, he always saw this ratio in the F2. This ratio can only come about if both parents here, both of these heterozygotes, form these four gamete genotypes in equal frequency. The other cross he did was to cross a double heterozygote F1 to a tester individual. And so homozygous recessive, green and wrinkled plants. When he did that, he always saw the 1 to 1 to 1 to 1 ratio, 25% of each offspring. Let's take a look at that cross, this one down here, up here, and chart it out on the Punnett square. If you do that cross, here are the gametes, right? That you're getting in equal frequency, 25% each. And this is the gamete coming from the tester individual. The offspring that you get are also going to be 25% each. 25% showing both dominant traits, 25% showing both recessive traits, 25% showing one dominant, yellow, and one recessive, wrinkled. 25% showing one, the opposite combination of one dominant and one recessive, green and round. Okay, so from this cross, 25% of showing each of the four different possible phenotypes. Of these phenotypes that are shown, some of these are non-recombinant or parental types. Yellow and round was seen all the way back here in the male parent, yellow and round. That's a non-recombinant or parental phenotype. Green and wrinkled, likewise, seen back here in parental generation, green and wrinkled. These two, however, are recombinant phenotypes. They're not seen in the parental generation. They're new combinations, recombinant. So yellow and wrinkled, green and round. Okay? For unlinked genes, when you do this cross, and this cross is called a dihybrid test cross. Dihybrid test cross. The results for unlinked genes from a dihybrid test cross always shows 50% recombinant offspring and 50% non-recombinant. Okay? That's what Mendel saw when he did this cross. So, this situation we're seeing here, when you do a dihybrid test cross, crossed a double heterozygote to a tester, you always see 50% of the offspring having recombinant phenotypes, not seen in the parental generation, 50% showing non-recombinant phenotypes. All right, So that's what Mendel saw for two genes that are unlinked. Now, how does that change for linked genes? Unlinked genes, and this is just basically repeating what we just saw on the video. Two genes, gene A determining one trait, gene B determining another trait. For a double heterozygote, right? It's heterozygous at both genes. All four possible allele combinations can occur, 1, 2, 3, 4, in equal frequency. And you see four different phenotypes. Two are parental, big A, big B. Seen back here, the yellow represents one parent. Little a, little b, also parental. Two of them are non-parental or recombinant types. Big A, little b, little a, big B. Okay? And so it's 50-50 parental or non-recombinant and 50% recombinant. How about for linked genes? So if two genes are linked together, they're physically attached. And so they're not acting independently. They're not going to assort independently. They tend to stay together. They would always stay together, except for the process of crossing over, which could separate them. So let's look at how linked genes behave during meiosis. And this is the standard cross that we will use. It's the cross that was in the video. It's called a dihybrid test cross. Dihybrid test cross is always going to be using an individual that's heterozygous for two genes. These are the two genes we want to know if they're linked or not. And the double heterozygote is crossed to a tester individual. So that's why it's called dihybrid, double heterozygote, test cross, tester is involved. In a dihybrid test cross, the tester individual will, since that individual is is homozygous recessive for both genes, that will not mask any of the phenotypes due to the double heterozygote. So we want to see what's going on in this double heterozygote during meiosis for genes that are linked versus unlinked. If two genes are linked, so this is showing A and B being linked, and it's also showing a double heterozygote. So if two genes are linked and no crossing over occurs during meiosis, that means the alleles that are linked here will stay linked. Meiosis will produce two types of gametes, and they are going to be both parental types. Okay? Both dominant here, that was a situation seen on this chromosome. Let's say that came from the mother. Both recessive here, that was a situation seen on the other chromosome. Let's say that came from the father. Okay? So for two genes that are linked, in which no crossing over occurs between those genes, there will be two different phenotypes seen, and they will be in equal frequency, 50% each. So in this situation, linked genes where no crossing over occurs, this is called complete linkage. Two genes are completely linked together and cannot be separated by crossing over. Okay, here's the situation if two genes are linked and crossing over does occur between those two genes. And this would happen, crossing over, during synapsis of prophase 1 of meiosis. If you remember, that's when homologous chromosomes pair and non-sister chromatids exchange pieces of the segments of the chromatid. So here's a crossover happening between two linked genes, A and B, in a double heterozygous individual. What will be the result? Well, we could just follow the chromatids here and the four types of gametes that would result from this meiosis. One gamete would have both dominant alleles, and that is a non-recombinant gamete. Non-crossover, non-recombinant. This one down here, little a, little b, same thing. Non-recombinant, it has the original combination, both recessive alleles. This one now, it's going to link a dominant A allele to a recessive B allele. This is a crossover gamete. It's a recombinant genotype, big A with little b. And likewise, this one down here, little a, is now going to be linked to big B, this gamete. Okay? So if two genes are linked and crossing over occurs, you will get 4 different types of gametes producing 4 different phenotypes in a dihybrid test cross. However, the non-crossover types, the non-recombinants, are going to be more frequent than the crossover types, the recombinants. And so the non-recombinant types, big A, big B, and little b, will sum to be more than 50%. And the crossover types, the recombinants, will be present at less than 50% sum. Okay? So that's kind of a general overview of the 3 possible outcomes of a cross. And this was summarized in a handout that I posted. So the next video, I'm going to go in a little bit more detail about those 3 scenarios. And we'll look at the expectations for genes that are unlinked, genes that are linked and no crossover can occur, complete linkage, and genes that are linked and crossover can occur. And by the way, that's called incomplete linkage. So I will stop the video here and see you in the next video.